Hi, I'm Sujin from University of Georgia. Today my topic was 3D root phenotyping system for field-grown maize. It is estimated that humans are responsible for releasing 880 billion tons of CO2 into the atmosphere. This actual CO2 has had serious consequences for the climate, such as global warming, extreme weather, and ocean acidification. Meanwhile, there's not enough carbon in the soil. Many of the world's cultivated soils have lost more than 50% of their original carbon stocks. The big question is, how can we reduce the actual CO2 in the air? The answer is literally right at our feet in the form of plant roots. Plants pull CO2 in from the air and turn into carbohydrate and sugars in the process of photosynthesis. Then they pump some of these sugars down through the roots to feed microorganisms who use that carbon to build healthy soil. Can you imagine? If we use this massive cropland with lots of maize roots as a natural carbon sink, how much CO2 can be pulled back to the soil? A study shows that 91 alien acres of maize reduced 815 million megatons of CO2. In that process, the deeper rooting maize varieties help to increase the soil carbon content by 1 gigatons. Now the question becomes, can we help breeders to select the best genotype maize to improve soil content through root phenotyping? To tackle this challenge, we developed a 3D root phenotyping system. We call that DIRT3D. DIRT3D uses a prototype of 3D root scanner to scan one excavated maize root in 6 minutes, and then compute its 3D root model. By tracking individual roots and building its root structure, DIRT3D was able to compute 18 root traits. Besides important elementary traits like the numbers, angles, and diameters of crown and base roots, DIRT3D was even able to compute traits like distance between walls, and which are hidden in the complex root structure. Since DIRT3D is an important step towards 3D root phenotyping, is it reliable and accurate? To answer this question, we validated DIRT3D on a test panel of 12 maize genotypes. The sample root images of each genotype and its 3D models are shown here. After validation, we got a promising result with coefficients of determination above 0.84 for all important traits. Given that DIRT3D can reliably measure 3D root traits, does it can distinguish genotypes? Here, we used principal component analysis method to analyze the variation of the 18 root traits. We can see that each genotype can be distinguished by a set of clustered points with same color, which indicates that DIRT3D can distinguish genotype. In summary, DIRT3D will enable breeders to select best genotypes to improve soil carbon content and save our planet from climate change. At last, we would like to thank the ARPA-E and NSF Career Grant that made this work possible, as well as our computational plant science lab at the UGA. Our DIRT3D manuscript preprint is also available online. Thanks for listening to my presentation.